For years, researchers searched for genetic clues buried in the lands where Moses once walked. Now, new ancient DNA results from the region reveal patterns no one expected. These findings do not just challenge familiar stories from the Bible, they open a door to secrets that may completely change how we understand Moses and the world he came from. The Bronze Age Canaanites. Step from the shadows. The Bronze Age in the Levant was a time of cities, trade, and cultural exchange. Archaeologists have uncovered evidence of thriving urban centers, pottery, tools, and burial sites that reflect a sophisticated society. One of the most significant discoveries came from Sidon, a city in modern-day Lebanon. In Sidon, researchers collected skeletal remains of 5 individuals who lived nearly 3,700 years ago. These remains were subjected to full genome sequencing, one of the most advanced methods in ancient DNA research. Scientists also sequenced the genomes of 99 modern Lebanese individuals to see how the ancient populations relate to people living today. The DNA analysis showed that the Canaanites themselves were not a homogeneous group. They were a mixture of local Neolithic populations who had lived in the Levant for thousands of years and migrants from regions in the east related to the Chalcolithic peoples of Iran. This blending of populations occurred long before the biblical period, suggesting that the region was already diverse and interconnected when the events described in the Bible were taking place. These genetic insights demonstrate that ancient populations moved, mixed, and interacted far more than the text might imply. Trade, migration, and family ties created a network of communities that shared ancestry across the Levant. This evidence challenges simple ideas of population replacement or pure lineages. The biblical narrative often presents the Canaanites as a people destined for destruction or displacement. In reality, the genetic data indicate that they were not completely removed from the region. Their genes survived in the people who came after them. This survival shows that the story of the land and its inhabitants was more complex than a single conquest or a dramatic erasure. The Canaanites adapted, integrated, and contributed to the ancestry of future generations. Modern populations in Lebanon carry these ancient threads of history in their DNA, showing a direct connection to a people who lived thousands of years ago. The discovery of this continuity also raises important questions about how we understand the biblical text. If the Canaanites remain present in the region, then the lines between the groups described in the Bible may have been more fluid than traditionally thought. The social and cultural boundaries were not as rigid as the stories suggest. This challenges readers to consider the past in a more nuanced way, where migration, interaction, and survival shape the history of peoples. If the Canaanites did not disappear and their descendants live among us today, then what does this mean for the other groups described in the Bible? Could other peoples whose stories are told as entirely separate actually share a common genetic heritage with their neighbors? Let's explore how genetic continuity affects our understanding of the biblical landscape and the connections between ancient populations and the modern peoples who inhabit the land today. Revelation in the DNA. The study of ancient DNA from the Levant reveals a remarkable story of continuity across thousands of years. Modern Lebanese populations carry more than 90% of their ancestry from Bronze Age Canaanite-related people. This shows that despite waves of invasions, migrations, and political changes, the core genetic makeup of the region remained largely intact. This continuity allows scientists to trace the threads of ancient life into the present and provides a window into the lives of people who lived during the time when the biblical stories began to take shape. The implications reach beyond the Canaanites. If one population thought to be erased survived and contributed genetically to modern peoples, other groups described in the biblical texts may also share unexpected connections. This includes early Israelites, Philistines, and other communities that appear in the narrative. The genetic evidence supports the idea that ancient identities were complex and fluid. The continuity revealed by the DNA also suggests that the biblical stories may have emphasized conflict and separation for narrative purposes, while in reality, the relationships among these peoples were more interconnected. Trade, marriage, alliances, and migrations shaped the region, creating networks that spanned cities and territories. This deeper understanding encourages a reevaluation of the social and cultural context of the Bible. It shows that the people of the Levant lived in a dynamic environment where identities overlapped and evolved. The genetic evidence shows a complex network of interactions is among neighboring peoples, raising new questions about identity, heritage, and continuity. Let's focus on the Jewish populations and the priestly Kohanim lineages, revealing how these ancient genetic patterns persist and how they connect to modern communities in ways that reshape our understanding of the biblical narrative. If foundational peoples in the region were already mixed before the biblical era, then what of the notion of a pure ancestry for the tribes of Israel, and what hidden lineages might still remain to be discovered? Jewish priesthood, the Kohanim lineages and genetic signatures. One of the most studied genetic lines connected to the Bible is the lineage of the Kohanim. These are the families who claim descent from the ancient priesthood of Israel. For many generations, this identity has been passed down from father to son. Scientists began to test the Y chromosomes of men who identified as Kohanim. The Y chromosome is useful because it is passed almost unchanged through the male line. When the results came back, researchers found something striking. A very large number of these men shared a common set of genetic markers that suggested they descended from a single ancient ancestor. This set of markers became known as the Cohen modal haplotype. It appears among Jewish men from different parts of the world. These communities had been separated for hundreds or even thousands of years, yet their Y chromosomes carried the same pattern. This created strong evidence that the priestly tradition preserved a real biological line from ancient times. The findings strengthened the idea that certain roles mentioned in the Bible may have been connected to genuine family lines, not only symbolic or chosen roles. More detailed studies later expanded this picture. Researchers discovered that the Cohen modal haplotype was part of a broader set of lineages that likely originated in the Near East. These lineages are common in Middle Eastern populations and also appear in non-Jewish groups from the region. This finding reveals that the priestly line is part of a much older genetic landscape. It reflects a population rooted in the Levant, long before written texts were formed. The ancient origins of this lineage link modern Jewish communities to the same world where the early Israelites lived and worshipped. The research also showed that not all Kohanim shared the same exact markers. Some had different but still related Y chromosome patterns. This indicates that the priestly line did not come from only one ancestor, but likely included several connected family lines that existed in ancient Israel. Over time, these lines may have expanded, moved, or blended with other groups, while still preserving the title and role of priesthood. This adds depth to the historical understanding of ancient Israelite society. It was complex, interconnected, and shaped by family traditions that survived even when populations migrated or were scattered. These findings also confirm that the region described in the Bible maintained strong continuity from ancient times to the present. The same genetic signals seen in the priestly line appear in other population sizes that lived in or near the Levant. This supports the idea that many groups described in the text shared deeper ancestral roots than previously thought. It also suggests that some traditions, such as the priesthood, may have been grounded in real historical structures. The DNA provides a traceable link between modern people and the ancient era that shaped the biblical narrative. If a priestly lineage can survive in the DNA for thousands of years, the question grows larger. What other lineages from the ancient Near East might still exist today, quietly carrying the history of the biblical world? Rereading biblical narratives in light of the genetics. The Bible places Moses in a very specific world. His story begins in ancient Egypt. During a time of strong centralized rule, he is described as being raised in an Egyptian household, then later fleeing into the land of Midian, which is associated with the northwestern Arabian region. From there, the narrative places him in the Sinai Peninsula before leading a population toward Canaan. These locations create a trail that stretches from the Nile Valley to the southern Levant. Each of these regions had distinct cultures, but their populations were connected through centuries of trade, migration, and shared ancestry. When scientists study ancient DNA from these lands, they find a picture that agrees with this interconnected world. People living in Egypt during the New Kingdom era carried ancestry linked to Northeast Africa, the Levant, and parts of the Near East. They were not isolated. Their genetic makeup reflected both African and Asian influences. Meanwhile, people in ancient Canaan carried ancestry rooted in the Bronze Age Levant, mixed with genetic signals from groups that had arrived earlier from regions near Iran and the Caucasus. The populations in Sinai and northwestern Arabia also carried blended ancestry, influenced by both the Levant and Arabia. This means that the world around Moses was full of genetic diversity, created by long-term regional exchange. These scientific results offer valuable context when thinking about the stories recorded in the Bible. The narrative often presents the Israelites as a community with a distinct origin. Yet the genetics of the region show that people living in these territories were already interconnected long before the time traditionally linked to Moses. It is likely that the people who followed him in the story were genetically similar to the broader Levantine populations. They would have shared ancestry with Canaanites, Egyptians, and neighboring groups. In other words, the lines drawn in the text represent cultural identity rather than clear biological separation. This connection becomes even clearer through ongoing genetic studies of ancient Canaanite samples. Modern Levantine peoples such as Lebanese communities, as well as many Jewish and Arab groups, still carry a large amount of Bronze Age Levant ancestry. This shows that the population did not disappear or get completely replaced by new arrivals. Instead, it stayed largely the same while absorbing new cultural and religious traditions. This continuity challenges the idea of total removal and suggests that the biblical descriptions of conquest and replacement may reflect symbolic storytelling rather than literal demographic change. The presence of mixed ancestry in ancient Levantine individuals also reshapes the way the biblical categories of insider and outsider are viewed. If the people of the region were already genetically blended, then the divisions described in the text must have been based on belief, law, and shared experience rather than biological origin. Genetic evidence supports the idea that identity in the ancient world was flexible. A group could adopt a new covenant or a new tradition without representing a separate population in genetic terms. These discoveries also change the way the movement of Moses and his followers can be imagined. If they traveled from Egypt into Canaan, they were entering a land filled with people who shared large portions of their ancestry. The genetic landscape does not show a dramatic difference between Egyptians of the New Kingdom and people living in the southern Levant. This means that any community forming under Moses would have blended naturally into the existing population. Ancient DNA cannot prove or disprove the events described in the Bible. However, it offers a valuable framework. It suggests that many of the groups described in the stories were closer to one another in ancestry than once thought. The genetic twist and the plagues. A surprising twist appears when researchers compare ancient DNA from the Levant with samples that have signals from North Africa and Kush. The admixture is real and measurable. It shows that some families who lived in the region carried ancestry linked to the Southern Nile and the lands beyond it. This is interesting because the Bible mentions that Moses had a Cushite or Ethiopian wife. The reference is brief but meaningful. It suggests that Moses moved in circles where African and Near Eastern identities met. His household was not defined by a single background, but reflected the diversity of the wider region. This leads to a new way of looking at the 10 Plagues passage. The story speaks of conflict between insiders and outsiders. It describes a people who are oppressed and another who rules over them. A social genetic approach offers another layer. The people living in Egypt at the time were not one single group. They were a mixture of local Egyptians and foreign populations who had settled there. Some came as servants, some as traders, and some through marriage or military alliances. This mixture could help explain tensions behind the narrative. A community with blended ancestry might be viewed with suspicion by the ruling class. Their loyalty could be questioned. Their presence could raise fears of alliances with foreign powers. The plagues can also be understood in a cultural context where identity was shifting. A population with mixed origins might follow new leaders more easily. Moses may have belonged to a lineage that linked him to both African and Near Eastern roots. If he carried ancestry similar to Cushite or Egyptian families, this would place him at a crossroads of identities. It may also explain why he had access to the Egyptian court yet chose to lead a community that saw itself as oppressed. His genetic background could have tied him to more than one world. The connection between North African ancestry and Levantine ancestry also opens the door to new interpretations. If Moses had ties to a Cushite line, he may have understood political dynamics beyond Egypt. Cush was a strong kingdom that sometimes allied with Egypt and other times opposed it. A leader with mixed ancestry might have been seen as a threat because he could form bridges between different populations. Such a figure might inspire loyalty among people who felt they did not fully belong to the Egyptian elite. This raises the most intriguing question of all. If Moses himself carried foreign blood, did this shape his role as a challenger of Egypt? Did he keep certain truths about his own ancestry hidden because revealing them could have changed the way others saw him? And could future genetic discoveries reveal even more about the origins of the man who transformed the history of an entire people? The future of biblical DNA research. What's next? The scientific study of ancient DNA has already changed our understanding of the ancient Near East, yet this field is still in its early stages. The next decade will bring an even deeper look into the world described in the Bible. Large projects in the Levant, Sinai, Jordan, Israel, and Egypt are now preparing to sequence remains from several periods that have never been studied before. These remains include individuals from the Iron Age, the centuries just before the Iron Age, and the early settlements that some archaeologists associate with emerging Israelite groups. Each successful sample brings us closer to understanding what the real population of that world looked like. Archaeologists are carefully selecting bones from many different layers of ancient sites. Some come from rural villages, others from fortified towns, and others from burial caves. A single well-preserved tooth or fragment of the inner ear bone can hold enough genetic material for analysis. Once these samples are sequenced, scientists can compare them with DNA from older Bronze Age groups and with modern populations. If the Iron Age samples show a clear break from earlier populations, that would support the idea of a major arrival or change. If they show continuity, it would suggest that people stayed in the region and that cultural identity changed faster than ancestry. Although the field is growing, major challenges remain. One difficulty is knowing exactly who the individuals were. A skeleton does not tell us if a person considered themselves an Egyptian, a Canaanite, a Judean, or an outsider from another land. Burial objects sometimes offer clues, but they are not always present, and they can be misleading. Another challenge is contamination. Bones from hot regions often lose much of their DNA, and modern handlers can leave tiny traces of their own DNA behind. Researchers must work in extremely controlled laboratories to make sure that the sequences truly belong to the ancient people being studied. A third challenge lies in interpretation. Genetics can show whether two populations share ancestry, but it cannot explain cultural identity. A person who carries ancestry from Egypt may have lived their entire life in Canaan and may have identified fully with the traditions of their local village. Cultural belonging does not always match genetic patterns. Scientists and archaeologists must work together to avoid reading too much or too little into the numbers. Even with these challenges, the broad picture is becoming clearer. The ancient world described in the Bible was not made up of isolated groups. It was a region filled with movement, trade, migration, and family ties that crossed borders. The people of the Levant, Egypt, Sinai, and Arabia shared ancestry in ways that earlier scholars could only guess. Although no DNA from Moses, Aaron, or other biblical figures exists, the population-level data already suggests that the world they lived in was genetically mixed and highly interconnected. Artificial intelligence is now adding a new dimension to this research. AI tools can read damaged genetic fragments and reconstruct missing pieces that once seemed impossible to recover. These tools can also compare thousands of genetic sequences in seconds and reveal patterns that human researchers might miss. In addition to DNA, AI is being used to read ancient scripts that were once considered unreadable. Burned scrolls, cracked tablets, and faint inscriptions are being converted into clear text through advanced imaging and machine learning programs. As AI helps decode unknown languages and reconnect broken stories, historians are gaining new insight into how ancient people described themselves and their world. This merging of genetics, archaeology, and artificial intelligence is creating a new kind of historical research. It may soon allow scholars to follow family lines across centuries, understand forgotten migrations, and uncover how cultural changes spread from one region to another. The next major breakthrough will likely come when scientists publish the first genome from a securely dated Iron Age Israelite context. When that moment arrives, what will it reveal? Will it support the traditional understanding of the ancient people, overturn long-held assumptions, or open an unexpected doorway into a story that has been hidden in the genes of the past for thousands of years? Would you trust ancient DNA and artificial intelligence to rewrite parts of history we thought were settled? Tell us your thoughts in the comments, because this story is only getting stranger.